In today's event, we are going to see the regulation of host gene expression in prokaryotes and eukaryotes. In the beginning, we will see about the regulation of eukaryotic gene expression system. Eukaryotic gene regulation, especially in multicellular organisms, is complicated by a process of development unique to the multicellular organism. Each multicellular organism begins as a single cell zygote which divides by mitosis. Cells differentiate into functional types by using some genes but ignoring others. Homeobox genes establish the body plan and position of the organs in response to gradients of the regulatory molecules. The timing of certain gene expressions seems to follow a sequence such as the production of different types of fetal hemoglobins by mammalian red blood cells, which switch to adult hemoglobin sometimes after birth. Clearly, the inactivation of certain genes occur in every adult cell. Therein lies the cure for cancer, old age, etc. The latest estimates of a eukaryotes are like this. If you see the latest estimates, there are about 21,000 genes in a human cell. Some of these are expressed in all cells all the time. These are called the housekeeping genes. These so-called housekeeping genes are responsible for the routine metabolic functions like respiration common to all the cells. Some are expressed as a cell enters a particular pathway of differentiation. Some are expressed all the time in only those cells that have differentiated in a particular way. For example, a plasma cell expresses continuously the genes for the antibody it synthesizes. Some are expressed only as conditions around and in the cell change. For example, the arrival of a hormone may turn on or off certain genes in a cell. There are several methods used by eukaryotes. One is the altering the rate of transcription of the gene. This is the most important and widely used strategy. However, eukaryotes supplement transcriptional regulation with several other methods. Altering the rate at which RNA transcripts are processed while still within the nucleus. RNA processing the pre-mRNA to mRNA. All the primary transcripts produced in the nucleus must undergo processing steps to produce functional RNA molecules for export to the cytosol. Most eukaryotic genes are split into segments. In decoding the open reading frame of a gene for a known protein, one usually encounters periodic stretches of DNA calling for amino acids that do not occur in the actual protein product of the gene. Such stretches of DNA, which get transcribed into RNA but not translated into protein, are called introns. Those stretches of DNA that do code for amino acids in the protein are called exons. Examples: the gene for one type of collagen found in chicken is split into 52 separate exons. Even the genes for rRNA and tRNA are split by introns. The human genome is estimated to contain some 180,000 exons, with a current estimate of 21,000 genes. The average exon content of one gene is about 9. In general, introns tend to be much longer than exons. An average eukaryotic exon is only 140 nucleotides long, but one human intron stretches for about 480,000 nucleotides. Removal of the introns and splicing the exons together are among the essential steps in synthesizing the mRNA. The steps of RNA processing. At first is the synthesis of the 5' cap. The synthesis of the cap is a modified guanine which is attached to the 5' end of the pre-mRNA as it emerges from RNA polymerase II, called the poly II. The cap protects the RNA from being degraded by the enzymes that degrade RNA from the 5' end, serves as an assembly point for the proteins needed to recruit the small subunit of the ribosome to begin translation. Secondly, it is the step-by-step removal of the introns present in the pre-mRNA and splicing of the remaining exons. This step takes place as the pre-mRNA continues to emerge from poly II. Next is the synthesis of poly A tail. This synthesis of poly A tail is a very important step. This is a stretch of adenine poly A nucleotides when a special poly A attachment site in the pre-mRNA emerges from the pol II. The transcript is cut there and the poly A tail is attached to the exposed 3' end. This completes the mRNA molecule which is now ready for export to the cytosol. The remainder of the transcript is degraded and the RNA polymerase leaves the DNA. Next step, the cutting and splicing of mRNA must be done with great precision. If even one nucleotide is left over from an intron or one is removed from an exon, the reading frame from that point on will be shifted, producing new codons specifying a total different sequence of amino acids from that point to the end of molecule, which is often ends prematurely anyway when the shifted reading frame generates a stop codon. The removal of introns and the splicing of exons is done by spliceosomes. These are a complexes of 5' snRNA molecules and some 145 different proteins. The introns in the most pre-mRNAs begin with a GU and end with an AG. Presumably, these short sequences assist in guiding the spliceosome. Next important step is altering the stability of messenger RNA. Altering the stability of mRNA molecule, that is, the rate at which they are degraded. RNA interference comes into picture here. RNA interference. In testing the effects of antisense RNA, one should use sense RNA of the same coding region as control. Surprisingly, preparations of sense RNA often turn out to be effective and inhibitor as antisense RNA. Why? It seems that the preparations of sense RNA often are contaminated with hybrids, sense and antisense strands that form a double helix of double-stranded RNA. Double-stranded RNA corresponding to a particular gene is a powerful suppressant of the gene. In fact, The suppressive effect of the antisense RNA probably also depends on its ability to form dsRNA, that is the double-stranded RNA, using the corresponding mRNA as a template. The ability of the double-stranded RNA to suppress the expression of a gene corresponding to its own sequence is called RNA interference, called the RNAi. It is also called the post-transcriptional gene silencing or PTGS. Mechanism of RNAi. The only RNA molecule normally found in the cytoplasm of a cell are the molecules of single-stranded RNA. If the cell finds molecules of double-stranded RNA, that is the dsRNA, it uses an enzyme called dicer to cut them into fragments containing nearly 21 base pairs of nearly 2 turns of a double helix. The 2 strands of each fragment then separate, releasing the antisense strand. With the aid of a protein, it binds to the complementary sense sequence on a molecule of mRNA. If the base pairing is exact, the mRNA is destroyed. Because of their action, these fragments of RNA have been named small interfering RNA or siRNA. The complex of siRNA and the protein is called the RNA-induced silencing complex, which is also called as RISC. siRNAs can also interfere with transcription. There is growing evidence that siRNAs can also inhibit the transcription of genes, perhaps by binding to complementary sequences on DNA or perhaps by binding to the nascent RNA transcript as it is being formed. In fission yeast, at least the siRNA is complex with one molecule of each of 3 different proteins. The entire complex is called the RITS complex. RITS stands for RITS, RNA-induced initiation of transcriptional gene silencing complex. How these siRNAs synthesized in the cytosol gain access to the DNA in the nucleus is unknown. Synthetic siRNA molecules that bind to gene promoters can, in the laboratory, repress transcription of the gene. The repression is mediated by methylation of the DNA and the promoter and perhaps methylation of histones in the vicinity. For example, there is a strain of rice LCG1 that produces abnormally low levels of proteins called gluteins. It turns out that of several glutein genes found in the rice, 2 closely similar glutein genes are located back to back on the same chromosome. In the LCG1, a deletion has occurred between the 2 genes, which removes the signal that would normally stop transcription after the first gene. Thus, RNA polymerase II transcribed right past the first gene and onto the second. The result is a messenger RNA which almost identical sequences running in opposite directions. That causes the mRNA to fold up into a molecule of double-stranded RNA. A dicer-like enzyme cuts up the double-stranded RNA into small interfering RNAs called the siRNAs that suppress further transcription of these genes as well as other glutein genes. altering the efficiency with which ribosomes translate the mRNA into a polypeptide. Protein coding genes have exons whose sequences encodes the polypeptides. Introns that will be removed from the mRNA before it is translated. A transcription start site, a promoter, a basal core promoter located within about 40 base pairs from the start site, upstream promoter which may extend over as many as 200 base pairs farther upstream, then it has enhancers, then it has silencers, then it has adjacent genes are often separated by an insulator which helps them avoid crosstalk between each other's promoters and enhancers. Then let's see the transcription start site. This is where a molecule of RNA polymerase II, Pol2, also known as RNAP2, binds. Pol2 is a complex of 12 different proteins shown in the figure in yellow with small colored circles superimposed on it. The start site is where the transcription of the gene into RNA begins. The code promoter. All eukaryotic genes contain a code promoter. One common example is a sequence of bases. Example TA, TAA, AAA, all called as Tata boxes. It is bound by large complex of some 50 different proteins including transcription factor 2D, TF2D, which is a complex of Tata binding protein TBP, which recognizes and binds to the Tata box, 13 other protein factors which bind to TBP, that is Tata box binding protein, and each other but not to the DNA. Transcription factor TF2B, which binds to the DNA, and Pol2. A code promoter with little variation in its structure and binding factors is found in all protein coding regions. This is in sharp contrast to the upstream promoters whose structure and associated binding factors differ from gene to gene. Many different genes and many different types of cells share the same transcription factor, not only those that bind at the core promoter, but even some of those that bind upstream, which turns on a particular gene in a particular cell is probably the unique combination of promoter site and the transcription factors that are chosen. Hormones exert many of their effects by forming transcription factors. The complexes of hormones with their receptor represent one class of the transcription factor. Hormone response elements to which the complex binds are promoter sites, and enhancers or some transcription factors called the enhancer binding protein binds to the region of DNA that are thousands of base pairs away from the gene they control. Binding increases the rate of transcription of the gene. Enhancers can be located upstream, downstream, or even within the gene they control. There are thousands of enhancers in the genome, but which ones are active depends on the type of the cell and the signals which it is receiving. Most genes, at least in Drosophila, are regulated by 2 to 3 enhancers, but some may be controlled by 8 or more. Multiple enhancers are particularly characteristics of housekeeping genes. One possibility is that enhancer binding proteins In addition to their DNA binding site, have sites that bind to transcription factors called the TF factors assemble at a promoter of the gene. This would draw the DNA into a loop as shown in the figure. Recent evidences shows that these loops are stabilized by cohesion, the same protein complex that holds the sister chromatids together during the mitosis and meiosis. Significance of looping. The looping of chromosomes that brings enhancers close to promoters and promoters close to other promoters seems to be a mechanism to ensure the expression or inhibition of the groups of the genes that must perform together. The response of a cell to the arrival of the signal, example a hormone, may involve turning on and off hundreds of different genes whose products must be produced in a coordinated way for the cell to respond appropriately. The dynamic movement of the portions of the chromosome carrying the appropriate gene loci into a transcription factor may be a mechanism to accomplish this. If so, we are seeing the eukaryotic equivalent of the coordinated gene expression provided by operons in the bacteria. We'll see how the role of silencers influence the transcription. Silencers are control regions of the DNA that, like enhancers, may be located thousands of base pairs away from the gene they control. However, when transcription factors binds to them, expression of the gene they control is repressed. Let's see insulators, how they control the expression. Insulators are the stretches of the DNA as few as 42 base pairs but may do a lot of trick. Located between the enhancers and the promoters or silencers and the promoters of adjacent genes or clusters of adjacent genes. Their function is to prevent a gene from being influenced by the activation or repression of its neighbors. Let's see how the mechanism goes in prokaryotes. For prokaryotes, the operon is a model system. Prokaryotes are sensitive to their environment and their genetic activity is controlled by the specific protein that interact directly with their DNA to quickly adjust to the environmental changes. Genetic expression is a process where genotypes coded in the genes are exhibited by the phenotypes of the individuals. The DNA is copied by the RNA and then synthesized into a protein. The process of transcription, which is synthesis of RNA from the DNA template, is where the regulation of the gene expression is most likely to occur. A difficult setting of prokaryote appears to allow for the continual synthesis of a protein to occur, whereas in eukaryotes the system is normally off until activated. An operon is a self-regulating series of genes that work in a concert. An operon includes a special segment of genes that are regulators of the protein synthesis but do not code for protein called the promoter and operator. These segments overlap and their interaction determines whether the process will start and when will it stop. RNA polymerase must create RNA by moving along the chromosome and reading the genes in the process of transcription. RNA polymerase first attaches to the promoter segment, which signal the beginning of a particular DNA sequence. If not blocked, it passes over the operator and reaches the protein producing genes where it creates the mRNA, then instruct the ribosome to create the desired protein. This process continues until the system is blocked by repressor proteins. Repressors bind to the operator and prevent the RNA polymerase from proceeding to create mRNA by prohibiting access to the remainder of the protein producing genes. As long as the repressor is binding to the operator, no proteins are made. However, when an inducer is present, it binds with the repressor, causing the repressor to change the shape and release from the operator. When this happens, the RNA polymerase can proceed with transcription and protein synthesis begins and continues until another repressor binds with the operator. A lac operon model is probably the most studied and well known. In bacteria such as E. coli, 3 genes are a part of operon that code for 3 separate enzymes needed for the breakdown of lactose, a simple sugar. A regulatory gene located before the operon continually makes repressor protein that bind with the operator and prohibit the function of RNA polymerase. The system therefore remains off until a flood of lactose molecules bind with all the available repressors and prevent their attachment to the operator. When the operator is free, the production of the enzyme to the breakdown of lactose continues until enough of lactose molecules are broken down to then release repressor to recombine with the operator to stop the production of the enzyme. 2 additional types of operons exist that operate in the same way except for the function of the operator. The TRP operon, the trip operon, differs because the repressor is active only when bounded to a specific molecule. For the remainder of the time, it remains unbounded and inactive in the absence of that molecule. Finally, in a positive twist, activators are used by a third type of operon to bound directly with the DNA, which allows the RNA polymerase to work more efficiently. Absence of the activators, RNA polymerase proceeds at a very slow rate.